Bismillahirrahmanirrahim. Today's topic is plasma protein. You have been gone through definition, classification, functions of proteins, different level of protein structure, and today's topic is plasma proteins. Plasma proteins are the most abundant molecules which are present in the circulation, and their normal concentration is from 6 to 8 grams per deciliter. The only component of the blood which is in the concentration of grams per deciliter. Now the learning objectives of this lecture is that at the end of the lecture you should be able to explain the difference between blood, plasma and serum. You should be able to define the role of plasma proteins and their broad classification, and then you should be able to discuss the structure and functions of immunoglobulins. Sometimes when we talk about the investigations, we use the word blood glucose, plasma proteins, serum creatinine. So you should be able to understand the difference between blood, plasma and the serum. Plasma is the fluid portion of the blood in which the formed elements are suspended, and we obtain plasma when the whole blood containing anticoagulant is centrifuged. So the supernatant of that centrifuged fluid is plasma, and the formed elements, they are being settled at the bottom. Plasma contains clotting factors. So, and if what is serum? Serum is the fluid portion of the clotted blood. Now remember, if we have to obtain plasma, we have to add anticoagulant into the blood sample, but for serum we let the blood to clot. So serum is fluid portion of the clotted blood. It doesn't contain form elements, that is RBCs, WBCs and platelets, and it also not contains plasma protein. And serum is obtained after centrifugation, centrifuging the clotted blood. So serum doesn't contain clotting factors. Now this picture shows you the different type of the plasma proteins. The major plasma protein is albumin, that constitutes the 60% of the plasma. And remember one thing, that albumin and clotting factors, they are produced by liver. So we can categorize plasma proteins into two categories: the one which are being synthesized by the liver, and the portion of the plasma which is synthesized by the bone marrow by the beta lymphocytes. So albumin is mainly synthesized by liver. Simultaneously, the clotting factors are the product of liver. It constitutes 60% of the plasma proteins, and its major function is to maintain colloidal osmotic pressure. Globulins, they constitute 35% of the plasma proteins. They are immunoglobulins, and globulins are also serve the transport function. Fibrinogen constitute 4%, and we know that fibrinogen has a role in the blood clotting. And only less than 1% of the plasma proteins are others, and like we will discuss them as we go on. Now this shows you the fractions of the plasma proteins: albumin and pre-albumin. Albumin we have discussed earlier. It is synthesized by the liver, constitute the 60% of the plasma. And pre-albumin is a protein which is tryptophan rich protein, and it has a short half-life. Then we have globulins. Globulins are again categorized into alpha 1, alpha 2, beta globulins and gamma globulins. The gamma globulins are synthesized by the plasma cells or the beta lymphocytes of the bone marrow. The alpha globulins include thyroxine binding globulin, transcortin. Transcortin is a protein, it is a globulin which is responsible for transportation of the corticosteroids. Then we have acid glycoprotein, alpha fetoproteins and alpha lipoproteins, and you can also see beta lipoproteins. So alpha and the beta lipoproteins has a role in the transportation of lipids in circulation. Lipids being water insoluble, they need protein for transportation into the circulation. Then among alpha 2 globulins we have haptoglobin. Haptoglobin proteins, they bind to the free hemoglobin, which is supposed to undergo degradation. So they bind with that hemoglobin molecule, and that hemoglobin molecule then undergo degradation, and you can have this ceruloplasmin. Ceruloplasmin is a protein which has two functions. Number one, it transport copper metal, and it also have a this ferrooxidase activity. It convert ferrous iron into ferric iron. We have already discussed this in the lecture of the iron metabolism. Then among beta globulins, we have transferrin, which is an iron transport protein. We have beta lipoproteins, which are responsible for the transport of lipids in the circulation, and C-reactive proteins. Now, C-reactive proteins are actually the proteins which are synthesized by the liver, and the levels of C-reactive proteins are increased. It means this somewhere in the body there is an inflammatory process. Among beta globulins, we have C3 and C4 components of the complement system. Now, this complement system is a part of the immune system, and this complement system enhances the ability of the antibodies as well as of the phagocytes to remove microorganisms and the dead tissues from the T cells, and also they have an ability to attack the plasma membrane of the pathogen. And then among gamma globulins, we have IgG, IgM, immunoglobulin A, immunoglobulin D, and immunoglobulin E. As far as these plasma proteins are concerned, these different fractions of the plasma protein can be separated out by applying various techniques. One of the technique is salting out method, and by this method albumin, globulins and fibrinogens can be separated out from the plasma proteins. And electrophoresis is another technique. In this technique there is a movement of the charged particles in a fluid or a gel which is subjected to electric field. So on an electric field the proteins are applied, and on the basis of their mobility they move towards the either anode or to the cathode. So it is the movement of charged particle in a fluid or gel under the influence of the electric. Now this is a normal pattern of this electrophoretic chart. You can see the albumin, they had the, as we have discussed earlier, the highest concentration, and then we have alpha 1, alpha 2, beta and gamma globulin fractions which are being shown in this. So this is normal electrophoretic graph of the blood plasma. Now this table also shows you that albumin, which is 60% of the total plasma proteins, synthesized by the liver, and it maintains colloidal osmotic pressure. It is not the only function that it serves. We will discuss the various other functions of the albumin simultaneously. Then globulins, which constitute 36%, alpha globulin, beta globulin, they are being synthesized again, as I have mentioned earlier, by the liver, and they have a role in the transport of lipids and that of the fat soluble vitamins. And gamma globulins, they have a role in the immunity, and the fibrinogens, which constitute 4%, again which is synthesized by the liver, has a role in the coagulation. Now this picture shows you that when the plasma proteins are within a normal concentration, and mainly the albumin, it retains water within the intravascular compartment. See, to retain water, 60% of our plasma is water, and this water should be retained within the intravascular compartment. Beside albumin, we have sodium and some osmotically active molecules which retain water within the intravascular compartment. So the upper part shows you that how the water is retained when the albumin is within the normal concentration, and due to any pathology, when the serum albumin concentration is low, the water move from intravascular compartment into the interstitial spaces, and you can see the patient is there's a edema is being developed. So what is plasma? Plasma is a liquid portion of the blood, and it is being obtained from the centrifugation of the anticoagulated blood, and 55 to 60% of the blood is made up of plasma, and plasma contains water, electrolytes, various metabolites like urea, uric acid, glucose, and it is a mean for transportation of the nutrients, hormones, immunoglobulins, and other proteins. And plasma proteins constitute approximately 4% of the total body proteins. Almost all plasma protein except immunoglobulins, they are synthesized by the liver, and they are generally synthesized on the membrane bound polyribosomes. Now if you recall, rough endoplasmic reticulum, which is an organelle, and it is being covered with multiple ribosomes, and ribosomes are the protein synthesizing factories. So this is why we are using the word polyribosomes. So ribosomes attached on the endoplasmic reticulum are the factories for the protein synthesis. And remember one thing, most of the plasma proteins are glycoprotein in nature. Albumin is purely this protein, but it doesn't contain. And in the lab, separation of the plasma protein can be done by salting out method. We have already discussed this. Now, the major function of the plasma protein includes, as we have discussed earlier, they maintain colloidal osmotic pressure. And the plasma proteins, mainly albumin, has a role in maintaining the pH of the blood, that is buffering action. Immunoglobulins, they serve the defense function. They have a role in the immunity. Coagulation factors like fibrinogen, plasminogens, they have a role in the coagulation of blood. They also maintain blood viscosity and the capillary permeability. And other important function, that is the transportation of nutrients, hormones, vitamins, and they are responsible for conservation of important elements. And most of all, they are the carriers, means of transportation of amino acid to the tissues. Once the protein is being taken orally, it undergo degradation in the GIT, and that proteins have been broken down into constituent amino acids, and these amino acids then they are being carried by the plasma to the liver, so that the liver can synthesize those proteins which are required by the body. Now this table shows you the various proteins which play a role in the transportation of molecules along with the ligand they transport. Now albumin, remember one thing, regarding albumin is that it constitutes the 60% of the plasma protein. Number two, it is highly negatively charged protein, so it binds with the divalent and trivalent cations. Definitely it is attracted towards the positive charge, so it binds with the copper and with the iron. And remember, copper and iron are transitional elements. They can change their valencies from cupric to cuprous, ferric to ferrous. Then we have another transport protein which discussed earlier, that is ceruloplasmin, which transport copper. Then in the circulation, the iron is being transported by the protein that is called as transferrin. Then we have certain globulins which bind with the hormones. Those hormones, especially which are steroidal in nature, which are derived from lipids, they bound to, because as I have told you earlier, the lipids are water insoluble, so they cannot be transported as such in the plasma. So they bind with the proteins. So globulins, they bind with the steroidal hormones, and they are the means of transportation of those steroidal hormones. Thyroid hormone, though it is being derived from its amino acid thyroxine, but it acts like a steroidal hormone, and in the circulation it binds to the globulin that is called as thyroid binding globulin. And thyroid hormones are T3 and T4, thyroxine and triiodothyronine. So these two forms of the thyroid hormone, they bind to the globulin. Then transcortin, as we discussed earlier, or cortisol binding globulins, they bind with the hormone cortisol. And sex hormone binding globulins, they bind with the hormones which have a role in this male and female secondary sexual characteristics like androgens, testosterone, estrogen, and the active form of the estrogen is estradiol. Then albumin binds with heme, bilirubin, and biliverdin. As we discussed earlier, haptoglobin binds with the free hemoglobin, which is supposed to undergo degradation. And as we have discussed earlier, fatty acid binding. So albumin binds to free fatty acids. When we say non-esterified fatty acid, it means fatty acid which is free, which is not attached to any and steroids. Now this table also shows you the concentration of these plasma proteins. We will mainly concerned with that of the albumin. You see, well, albumin, as I told you earlier, the plasma proteins are the only fraction of the blood which are present in grams per deciliter. Otherwise, other components of the blood are present either in the milligram concentration or millimole concentration. So albumin, the normal concentration of albumin is 3.5 to 5 grams deciliter, and it binds and it transport free fatty acids, bilirubin, calcium, a number of drugs. They bind to the this albumin for transportation in the circulation. And as we have discussed earlier again, that it maintains colloidal osmotic pressure. Prealbumin, that is responsible for the transport of thyroxine to some extent. And then alpha 1 globulin, they are in a concentration of 0.3 to 0.5 grams per deciliter, and they combines with elastase, trypsin, and protect the hydrolytic damage of the tissue such as the lung. Then retinol binding protein, vitamin A is present in the body in 3 different forms: retinol, that is alcohol form, retinal, and retinoic acid. So retinol binding protein is responsible for the transport of vitamin A, and it is in a concentration of 3 to 6 milligram per deciliter. Thyroxine binding globulin, we have just discussed earlier, they transport thyroid hormone. Then we have cortisol binding proteins. We have already discussed these functions, so I will just go for their plasma concentration. Transportin is 3 to 4 milligrams per deciliter. Now, as far as you people are concerned, you should know that what is the total plasma protein concentration? It is 6 to 8 grams per deciliter. Out of this 6 to 8 gram per deciliter, 3.5 to 5.5 is the albumin. Remaining are the globulins and other clotting factors. Okay, so you are not supposed to remember the plasma concentration of transcortin or ceruloplasmin or haptoglobulin, just total plasma protein concentration, albumin concentration, and the globulin fraction, how much it is. So these are plasma concentrations of various proteins which have a role in transportation. And if you see at the end, CRP, the C-reactive proteins, which I told you earlier that it is being synthesized by liver. And again I will repeat, plasma proteins are broadly classified into 2 classes: the one fraction of the plasma protein which are synthesized by the liver, and another fraction is synthesized by the bone marrow, by the plasma cells or the beta lymphocytes. So C-reactive proteins are produced in the liver as a result of inflammation anywhere in the body, and its levels are indicated of the acute inflammation. And C-reactive proteins, they motivate phagocytosis in the inflammatory disease. Here we will discuss the abnormal protein pattern on electrophoresis in some clinical diseases. Like in chronic infections, there will be peak on the gamma globulins, and the multiple myeloma, which is a tumor of the plasma cell, there will be a sharp spike of M band between the beta and the gamma, and immediately after this slide we will see the electrophoretic pattern. In primary immune deficiency, the gamma globulin fraction is reduced. In nephrotic syndrome, the fraction which is being shown increase is that of the alpha 2 fraction. Alpha 2 fraction will show spike, and in the cirrhosis of liver there are some fast moving gamma globulins that prevent resolution of the beta and the gamma globulin bands. So you can see three patterns are shown in front of you. In the normal, we can see there's a spike showing you the albumin, and then we can see alpha 1, alpha 2, beta and gamma globulins in the normal pattern, and you can compare them with that of the inflammatory conditions and in the multiple myeloma. Now, as far as the multiple myeloma is concerned, it is a tumor of the plasma cells, and plasma cells synthesizes immunoglobulins, which are called as paraproteins, and these paraproteins, they don't play any role in the immunity. Now in the inflammatory process, you can see a spike in the alpha 2 fraction, and you also see the rise in the beta globulins. And in the multiple myeloma, you can see a high spike greater than that of the albumin of that of the gamma globulins, and these are the paraproteins. And as I told you, paraproteins are the fraction of the globulins which have no role in immunity. Albumin, the major plasma protein. Its molecular weight is 69,000 Dalton, and it is comprised of a single polypeptide chain of the 585 amino acids. If you recall, in the last lecture I told you all polypeptides are protein. Okay, so and it has 17 disulfide bonds, and it is highly negatively charged at the physiological pH. So on the electrophoretic field, it moves towards the cation because of this negative charge, and because of this negativity, it is responsible for the transportation of the transitional elements like copper, iron, and it constitutes 60% of the plasma proteins. Half-life is 20 days. As far as the secondary structure of protein of the albumin is concerned, more than 50% in the alpha helical structure, and approximately 15% of its chains are in the form of the beta pleated sheets, and remaining is in the random coil conformations. So as far as the tertiary structure is concerned, it is a globular protein. So we can even code. If somebody asks you about the example of a tertiary structure of protein, you can say beside myoglobin that albumin is an example of the tertiary structure of protein. Now here are various functions of albumin. We have been through these previously, but again, nearly 80% of the colloidal osmotic pressure is maintained by the albumin. Beside that, we have some osmotically active molecules, like as I told you earlier, sodium. We have glucose, which is osmotically active molecule. So these also retain fluid within the intravascular compartment. And then its another important function is transportation. So it is responsible for the transport of bilirubin free fatty acids, phospholipids, cholesterol, amino acids, steroidal hormone, thyroxine, and a number of drugs like sulfonamide, aspirin, salicylates, and cations like calcium, copper, iron, and being again the it is a contributor of it maintains the pH of the blood by its buffering action. And nutritional function, it's mainly proteins are meant for the bodybuilding, not for the nutritional purpose. But when the body is deprived of its primary source of carbohydrates and then lipids, then it do serve the source of amino acids in tissues. This is its function, that it again it has a role in the tissues for amino acids. Okay, so what happens? Hypoproteinemia. Hyperproteinemia is a condition. Mia means blood. So hypo means less protein in the blood. Normal concentration of the plasma protein is 6 to 8 grams per deciliter. So if the concentration is less than 6 grams per dl, then we'll term it as hyperproteinemia. And mainly the albumin fraction is one which is mostly affected. And the causes of the hyperproteinemia: prolonged starvation, that is the malnutrition, malabsorption from the GIT, and if, as we have discussed earlier, that plasma proteins mainly categorized into two categories: the one which is synthesized by the liver. So if the liver is non-functioning due to any reason, due to cirrhosis of liver, due to hepatitis, or due to the any pathology, then liver will not be able to synthesize protein as required by the body. And then liver is capable of synthesizing protein, but the body is losing that protein in the urine, like in nephrotic syndrome. So nephrotic syndrome, cirrhosis of liver, defective absorption from the GIT, and prolonged starvation are the main reasons for the hyperproteinemia. And what happens when the protein concentration is decreased? What will happen? The colloidal osmotic pressure will decrease. Water will diffuse from the intravascular compartment into the interstitial compartment, and the individual will develop generalized edema. If the globulin fraction is decreased, definitely the immunity is compromised. And similarly, the fibrinogen and clotting factors which are being synthesized by the liver, their synthesis will also be affected, and the bleeding tendency will be increased in the affected individual. Now this slide shows you the various fractions of the globulin, and the one thing which you must remember, that except gamma globulins, the alpha and the beta globulins are synthesized by liver. We have already went through this description of the alpha and the beta globulins, and in the next slide we will discuss their. This slide is simply the recall of the previous one, that globulins are mainly classified into three classes, that is alpha, beta, and gamma. And their molecular weight is approximately 156,000 daltons. Alpha and the beta globulins synthesized by the liver, and mainly they are involved in the transport function. And you can see that these alpha and the beta globulins, they carry substances like thyroid hormone, cholesterol, those hormones which are steroidal in nature like cortisol, aldosterone, and then free fatty acids, iron, insulin, and fat soluble vitamins like vitamin A, D, and K. Must remember that most of the clotting factors are either alpha or the beta globulins. You have heard this word angiotensinogen, if you recall, then when we discuss peptides. Angiotensinogen is a protein, and and as we know that most of the peptides, they derive from the breakdown of the proteins. So angiotensinogen is converted into angiotensin 1 and 2, which are deca and the octapeptides, and which have a role in maintenance of the blood pressure. Now this slide will show you the electrophoretic pattern of the various plasma proteins. You can see various spikes, and we have already gone through the albumin, so we'll here see that examples of this alpha 1 globulins is antitrypsin, thyroid bonding globulins. Then you can see a spike that has been showing you the alpha 2 fraction of this globulin, and it includes haptoglobin, ceruloplasmin, and alpha 2 macroglobulins. Then beta globulins includes transferrin and beta lipoproteins. Transferrin is an iron transport protein, and beta lipoproteins are involved in the transportation of the lipids like LDL. And then we can see a spike that is showing us the gamma globulins, which are the immunoglobulins and which are synthesized by the plasma cell. Immunoglobulins. Immunoglobulins are also known as antibodies. And remember, these immunoglobulins are glycoprotein in nature, and they have a very critical role in the immune response because they have a specificity that antibodies can recognize and bind particular antigens such as bacteria and viruses, and then they facilitate their destruction. Now this slides will show you the 5 different type of immunoglobulins: immunoglobulin G, immunoglobulin M, A, IgD, and IgE. Immunoglobulin G is one of the immunoglobulins which is being transferred from mother to the fetus and the newborn. Immunoglobulin A is mainly present in the various secretions, and immunoglobulin M is the first antibody or the immunoglobulin which is produced in the body as a response to the exposure to the foreign antigens. Immunoglobulin E is a mediator of allergic phenomena, and immunoglobulin D, its function is not being so far specified. As we just discussed that immunoglobulins are classified into 5 different classes. They are being distinguished on the basis of 2 things: the type of the heavy chain they contain and the unit they are comprised of. Now IgD, IgE and IgG, they are comprised of monomeric units, where you can see that IgA has the 2 units. It's a dimer. And as far as the structure is concerned, that of the IgM, it is complex and it is comprised of 5 different monomeric units. It is a pentamer. This slide shows you the basic structure of all immunoglobulins. You can see here the source is the plasma cell, which are the cells of the bone marrow, and in response to exposure to the foreign antigens, whether it can be a bacteria, it can be a virus, it produces immunoglobulins. The basic structure of this immunoglobulins, it is comprised of 2 identical heavy chains, and simultaneously it also has the 2 identical but smaller in size, the light chain. This is the extensive version of this immunoglobulins. You can see that the amino terminal of both the heavy and the light chain, they have a antigen, they form a pocket, and that pocket is a site for the binding of the antigens. So antigens, when bind with this variable region of this light and the heavy chains, they form antigen antibody complex. Here we will discuss in detail the structure of the immunoglobulins. Immunoglobulins are V-shaped structure, and these are made up of 2 identical polypeptide chains which are called heavy chains, and simultaneously they also have 2 identical but in smaller size polypeptide chains which are called light chains. The structure of immunoglobulins, you can see in this slide, it has been stabilized by a number of the disulfide bonds, and the amino terminal of this heavy and the light chain is comprised of a variable amino acid sequence. These amino terminals of the light and the heavy that's why it is called as the variable region, and the light and the heavy chains, they amino terminals, they form a pocket which is for the binding of the antigen. That's why this region is called V region, or it is also called as the FAP, that is the antibody recognition region. Now the type of 5 different types of the heavy chains exist, and these heavy chains determine the class of the immunoglobulins. Heavy chains can be of alpha, gamma, delta, micro and epsilon, whereas the light chain has only two varieties, that it is comprised of the either kappa and lambda. Now the region of the heavy chain adjacent to the variable region, it is has a less variability in the amino acid sequence. That's why it is called as the FC region or the constant region. Now what happens when the antigen bind to the pocket in the variable region, conformational change taken place, and that conformational change is then transmitted to the FC region, and this FC region is then activated and start activating the complement system. This slide is giving us the details of the various types of the immunoglobulin, the type of the structure they have, where they are being, their location, and the function that is being served by that immunoglobulin. Now you can see the first immunoglobulin is IgG. It is a monomeric unit, and its location, it is being located in the extracellular fluid. The function of this IgG is to neutralize toxins, and it activates the complement system. As far as the structure of the IgA is concerned, it is a dimer, and it not only it is present in the extracellular fluid, but it is also present in the various mucous secretions, and it has an antibacterial activity. Immunoglobulin M, which has got the most complex structure, that is comprised of the 5 different units. It is a pentamer, and it is mainly present intravascularly, and it is the first antibody which is produced by the body in response to the infection. IgD, it is a monomeric unit. It is comprised of monomeric unit, and it is present on the extracellular fluid as well as on the cell membrane, but its function is not so far being identified. IgE, again a monomeric antibody, and it is present in the extracellular fluid, and it is being produced in response to either the parasitic infections or as a response to the allergic phenomena. Now we will discuss individual immunoglobulins. IgG. 75% of the antibodies which are present in the serum, they are of IgG type, and it is the most common type of antibody found in the circulation. Toxins and pathogens need to be neutralized, and therefore what happens, the IgG bind to these toxins and pathogens and clear them from the circulation by activating phagocytosis and their degradation. As far as concerned, Ig have 4 major effector functions, that is neutralization, opsonization, complement fixation, and antibody dependent cell mediated toxicity. You have been through the topic of immunity by the physiology department, so you must be familiar with all these terminologies. Now IgG is, as I told you earlier, it is the antibody which is being transferred from mother to the fetus and the newborn, and whenever we determine or estimate the levels of immunoglobulin G in the circulation, and if they are being increased, they indicate that either it is because of the allergies, it indicates the presence of the chronic infection, or autoimmune disorders such as the rheumatoid arthritis and the systemic lupus erythematosus, or the individual may suffer from the celiac disease. The secondary IgG deficiency may be caused by aging, malnutrition, chemotherapy, and the long-term use of the corticosteroids, or the infections like HIV, that is the human immunodeficiency virus syndrome. Now, what could be the possible signs and symptoms of the IgG deficiency? Nowadays, it is a pandemic of coronavirus, and you must have heard that those individuals who are immunocompromised, or immunocompromised means those who have this immunoglobulin deficiencies. They are the victims of this virus infection. And those individuals which have IgG deficiency, they may present with the sinus infections or other respiratory infections. Their GIT may be involved. They may show ear infections, pneumonia, bronchitis, or infections that result in the sore throat. Rarely it can be it can lead to the life-threatening infections. The life-threatening infections are those which can cause death. Now, in some people, the deficiency of IgG results in the infection that causes the scarring. Or you must be familiar with the word fibrosis. So the scarring or fibrosis affects the airway and the lung function, and the patient may develop breathing problems. And the people who are IgG deficient, even those vaccines for pneumonia and flu doesn't keep them getting these infections. The second immunoglobulin is immunoglobulin M. And as I told you, that when the body gets exposed to any kind of infection caused by bacteria, viruses, or any other infecting agent, body starts making antibodies which are of IgM type. So it is the body's first line of defense against infection. So remember, the first antibody produced by the body in response to infection is IgM. So it indicates acute infection. Acute infection means infection which is of sudden onset and of brief duration. Now, if you recall, it has got a complex structure. It is the pentamer, and its large size confers the excellent binding specificity and avidity, so that it can pick up even the trace amount of the infection, so that this infection can then be phagocytes, or it can be removed from the body. So when the body senses any invader, IgM level rises for a short time, and then gradually it begins to drop as IgG comes into effect, and then there is a rise in the IgG, and IgG protects the body for a longer period. High levels of the immunoglobulins indicate that the individual might be suffering from heteroglobulinemia or early viral hepatitis, infection mononucleosis, rheumatoid arthritis, nephrotic syndrome, or it indicates the presence of a parasitic infections. Individuals with the low levels of IgM, they found difficulty in fighting the infections. Immunoglobulin A is unique in that sense that it prevents the mucous membrane of various organs and tissues from the infection. And IgA antibodies are found in the mucous membranes of lungs, sinuses, stomach, and intestine. And the amount of the IgA produced by these mucosal membranes is greater than all other types of the antibodies combined. So as far as the function is concerned, it is the principal antibody in the secretions that bathes mucosal surfaces, and it is the first line of defense against infection. It prevents all mucous membranes lining the lungs, GIT, stomach, and the intestine. So people who are suffering from the IgA deficiency, they experience pneumonia, ear infection, sinus infections. They are victims of allergies, asthma, and diarrhea. Another type of immunoglobulin is IgE. IgE is being claimed as a mediator of immediate hypersensitivity reactions, and it is involved in all allergic diseases like asthma, hay fever. Now what happens, this IgE, when it binds with the allergen, it triggers the release of histamine. It is histamine is an allergic mediator, and this histamine release is responsible for the wheal and the flare skin reaction which is observed in case of allergies. Now this pictures is showing you that how the histamine is being released from the mast cells. What happens that antibodies like IgE, toxin and drugs, they bind to the receptor on the mast cells, and from they trigger the release of the histamine. Histamine is a mediator of allergy, and when histamine is released into the circulation, it is responsible for the sign and symptoms which is observed in asthma or affecting the GIT, and may be responsible for diarrhea and other symptoms. IgE is also a defense against the worm infections. Worm infections are called helminths, and it prevents from this worm infections by causing the release of enzymes from the eosinophils. Now, while discussing the immunoglobulins, I have repeated a number of times that the immunoglobulins are produced by the plasma cells. Now, when the tumor of the plasma cell is called multiple myeloma. So what happens in multiple myeloma, the number of plasma cells are increased. So they start producing immunoglobulins which cannot fight infections. And these type of immunoglobulins are called paraproteins or M proteins. Now this this slide is a brief review of all types of immunoglobulins we have discussed so far, and their serum levels. You can see the highest concentration of the immunoglobulins which are present in circulation, that is 85%, are IgG. Then we have 17 to 15% IgA in circulation. Then comes the immunoglobulin M, which are 5% of the serum immunoglobulins. IgD is only 0.3%, and IgE is 0.02%. And we have already discussed that IgD, IgG and IgE are monomeric, whereas the IgA is a dimeric polypeptides, and IgM has pentameric structure. And we have already discussed these functions in details earlier, but you can see that these immunoglobulins, they are further subclassified, like in IgG, if you go for IgG1, IgG2, IgG3 and IgG4, depending upon the type of the heavy chains they contain. So as I discussed earlier, that heavy chains have 5 different type of these chains, polypeptide chain. They may be alpha, delta, gamma, delta, micro and epsilon. So the presence of the heavy chains in different is responsible for the subclassification of these immunoglobulins.